Imagine a microscopic machine booting up, but instead of silicon circuits, the hardware is a living cell, and the operating system is a genetic sequence typed on a laboratory computer, compiling a sequence of adenine, thymine, cytosine, and guanine. This is not a scene from a distant science fiction future, but a reality unfolding in laboratories today. We have crossed a threshold where the code of life is no longer just something we read, but something we actively write. Th- this discipline is known as synthetic biology, a field often explained as the application of engineering principles to living systems. While earlier techniques involved cutting and pasting existing genes, a process akin to editing a document, this new frontier involves engineering biological systems and designing entirely new organisms from scratch. It treats DNA as a programmable language to construct artificial life that has never existed in the natural world, aiming for precise control over biological behavior. The watershed moment for this science arrived when genetic pioneer Craig V. Denter and his team unveiled the first synthetic cell, a milestone that forever blurred the line between the organic and the manufactured. They did not just tweak an existing bacterium, they sequenced a genome, synthesized it chemically in a lab, and transplanted it into an empty host cell. When that synthetic DNA booted up, it hijacked the cellular machinery and began to replicate, proving that life could be manufactured from digital information. Central to this monumental achievement was the concept of a minimal genome. Researchers systematically stripped away genes from a simple bacterium to discover the absolute minimum number of genetic instructions required for a cell to survive and reproduce. By reducing life to its barest essentials, they created a blank biological canvas, a foundational chassis upon which entirely new functions could be programmed and deployed with unprecedented accuracy. That foundational chassis shattered our traditional understanding of biology. If a living organism can be reduced to a digital file, transmitted across the globe via the internet, and printed into biological reality, the boundary between the organic and the digital dissolves. We are forced to confront the profound reality that life is fundamentally an information processing system, one that we are now learning to manipulate at its most fundamental level. The potential applications of these programmable biological machines are staggering, particularly in the realm of human health. Scientists are designing synthetic immune cells programmed to hunt down specific cancer markers, deliver targeted payloads, and then self-destruct once the tumor is eradicated. These living medicines represent a paradigm shift, moving away from static chemical treatments to dynamic, intelligent therapies that adapt to the patient's body in real time. Beyond medicine, this technology examines how we might completely overhaul global manufacturing and logistics. Imagine towering vats of engineered yeast that do not brew beer, but instead secrete spider silk, jet fuel, or biodegradable plastics. By harnessing the unparalleled efficiency of biological synthesis, we could replace polluting chemical factories with clean, renewable biomanufacturing facilities, fundamentally altering the industrial landscape and offering a vital tool in the fight against climate change. Yet, programming biological circuits is vastly more complex than writing software code. Cells are noisy, unpredictable environments driven by fluid dynamics and chaotic molecular interactions where genetic instructions can mutate, degrade, or interact in unforeseen ways. Engineering reliable biological parts that behave consistently across different generations of a cell remains a monumental hurdle, requiring a deep understanding of cellular dynamics that we are only just beginning to unravel. This unprecedented control over living matter inevitably triggers intense debate, often crystallized in the controversial accusation of playing God. When we design organisms tailored to our specific economic needs, we are assuming the role of natural selection, raising profound questions about the moral status of artificial life. The ethics of creating, patenting, and potentially exploiting bespoke life forms forces us to reevaluate our relationship with the natural world and our responsibilities as its architects. Perhaps the most terrifying shadow cast by this innovation is the specter of biosecurity. As the cost of DNA synthesis plummets and the technology becomes more accessible, the barrier to entry for creating weaponized pathogens dramatically lowers. The same tools used to engineer a cure for cancer could theoretically be hijacked to design a highly contagious, vaccine-resistant virus, presenting an existential threat that traditional national security apparatuses and border controls are entirely ill-equipped to handle. Leading security analysts warn that the democratization of biotechnology is vastly outpacing our regulatory frameworks. While legitimate laboratories operate under strict oversight, the proliferation of desktop DNA synthesizers and open source genetic databases creates critical vulnerabilities. Developing robust international protocols to monitor the synthesis of dangerous genetic sequences is universally recognized as an urgent priority, yet achieving global consensus remains a daunting political and logistical challenge. The risks extend far beyond intentional malicious use, bleed, bleeding into the realm of unintended ecological consequences. If a highly optimized synthetic microbe designed to clean up oil spills is released into the ocean, it could outcompete naturally occurring bacteria, disrupting the delicate balance of the marine food web. Once a self-replicating organism is introduced into the wild, recalling it is virtually impossible, making every environmental deployment an irreversible planetary experiment. To mitigate these environmental risks, researchers are developing sophisticated biological containment strategies, often referred to as kill switches. These genetic circuits are designed to trigger cellular death if the organism escapes its intended environment, such as a controlled bioreactor. Another approach involves engineering organisms to depend on synthetic amino acids that do not exist in nature, ensuring they simply starve to death if they escape into the wild. The ghost of those early containment debates lingers, however, because life is notoriously adept at finding a way to survive. Mutations can disable genetic kill switches, and horizontal gene transfer could potentially allow synthetic traits to migrate into wild populations. Ensuring the absolute evolutionary stability of these biological safeguards over millions of generations is a mathematical and biological puzzle that continues to confound the brightest minds in the field. Meanwhile, the agricultural sector is bracing for a massive disruption driven by engineered crop. We are moving beyond simple pest resistance toward plants designed to synthesize their own nitrogen fertilizer from the air, potentially eliminating the need for environmentally damaging chemical runoff. While this promises a revolution in sustainable farming, it also threatens to consolidate control of global food supplies into the hands of a few biotechnology conglomerates holding the patents. This economic shift highlights a deeper inequity in the emerging bioeconomy. If the synthetic alternatives for vanilla, rubber, or palm oil become cheaper than their natural counterparts, millions of farmers in developing nations face catastrophic economic displacement. The transition to a biomanufactured world must carefully navigate these socioeconomic fault lines, ensuring that the benefits of artificial life do not come at the cost of devastating human livelihoods and regional stability. Looking further ahead, the marriage of synthetic biology and deep space exploration opens mesmerizing possibilities. When humanity establishes a presence on Mars, we will not transport heavy construction materials or chemical factories across the solar system. Instead, we will send digital genetic codes, printing bespoke microbes designed to extract minerals from Martian regolith, generate breathable oxygen, and manufacture rocket fuel in the extreme radiation of an alien world, fundamentally solving the payload bottlenecks of interplanetary colonization. This extraterrestrial potential is being exponentially accelerated by cutting-edge artificial intelligence. Machine learning algorithms are now capable of predicting complex protein folding and designing novel genetic sequences that no human mind could conceptualize. By pairing the generative power of AI with the physical output of biological synthesis, we are creating a closed-loop system of automated evolutionary design, vastly compressing the timeline of biological innovation. As algorithms begin to author the code of life, the distinction between biological evolution and technological progress vanishes entirely. We are witnessing the birth of a new branch on the tree of life, one driven not by random mutation and natural selection, but by artificial intelligence and human intent. This engineered evolution fundamentally challenges our definition of what is natural, forcing us to accept that technology itself has become a biological force. Prominent futurists argue that this synthesis of silicon and carbon represents the ultimate technological singularity. The ability to program matter at the molecular scale, using the machinery of life, offers a pathway to a post-scarcity civilization. If we can grow our buildings like trees, synthesize our food from sunlight and ambient air without slaughterhouses, and program our bodies to resist aging, the fundamental constraints that have governed human history will be entirely rewritten. Yet realizing this utopian vision requires navigating a minefield of public perception and deep-seated fear. The concept of creating life in a lab inherently triggers a visceral, often negative reaction, heavily influenced by decades of dystopian science fiction. Building public trust requires transparent scientific discourse, acknowledging the valid fears surrounding biosecurity and ecological disruption, while clearly communicating the rigorous safety protocols being developed in tandem with the technology. The core tension lies in the asymmetry of risk and reward. The potential to cure genetic diseases and halt climate change is weighed against the existential threat of an engineered pandemic or a catastrophic ecological collapse. Deciding who gets to calculate that risk and who bears the consequences if those calculations are wrong is an ethical burden that cannot be solved by science alone, requiring deep democratic engagement and unprecedented global cooperation. Even with global cooperation, the sheer pace of innovation continually outstrips our capacity to fully comprehend its implications. Every new synthetic pathway we design, every artificial protein we fold adds a new variable to the incredibly complex equation of planetary biology. We are attempting to master a 4 billion-year-old operating system in a matter of decades, a breathtaking display of hubris that demands an equal measure of profound humility. Ultimately, the story of synthetic biology is the story of humanity's transition from passive observers of nature to active participants in its design. We have long shaped our environment through agriculture and industry, but we are now reaching into the very source code of existence. This power to dictate the future of biological form is the most profound capability our species has ever acquired, eclipsing even the splitting of the atom. As we stand on the precipice of this engineered genesis, the choices we make today will echo through the geological record. The tools to build a sustainable, disease-free world are being synthesized in the laboratory right now, alongside the tools of our own potential destruction. Our survival no longer depends merely on adapting to the natural world, but on the wisdom, restraint, and foresight with which we design the artificial one.